In this video, I will be talking about how DNA is converted into protein. DNA is made up of molecules called nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. The four types of nitrogen base are adenine, thymine, guanine, and cytosine. Order of these bases is what determines DNA's instructional or genetic code. In order to understand this process, we have to learn about transcription and translation. The mRNA formed in transcription is transported out of the nucleus into the cytoplasm to the ribosome. The process by which mRNA directs protein synthesis with the assistance of tRNA is called translation. RNA polymers with the main enzyme involved in transcription. It reads one of the DNA strands and adds complementary nucleotide bases to make an mRNA transcript. The mRNA will later be translated into protein. RNA polymers with the help of proteins called transcription factors binds. Transcription factors bind within a specific sequence within the gene, which is called the promoter. The promoter plies the two strands apart. One of the strands will serve as the template strand, which is used to generate the mRNA, and the other is the non-template strand. RNA polymer does not need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene and in a process called elongation, synthesizing the mRNA as it goes. Reading the antisense strand to 3' and 5' and generating the mRNA to the 5' and ending attaching RNA nucleotide to 3' end as it goes. Once RNA polymerase reaches the gene, termination occurs. The enzyme detaches from the gene and the DNA is returned to its original state, which produces an mRNA that carries the code in the gene. The enzymes will leave the nucleus where all the genetic materials or chromatin is and move into the cytoplasm, where it will find the ribosome. This is where translation occurs. During translation, mRNA acts as a code for a specific protein. During translation, mRNA acts as a code for a specific protein. This happens because each set of 3 bases on an mRNA, which we call codons. Codons will code for a specific anticodon, which will be carried by a specific transfer known as tRNA. Each tRNA is covalently linked to a particular amino acid. Since there are 4 bases, each codon has 3 letters. 4 to the root 3 equals 64 different possible codons, which is more than enough to code for all the amino acids we need. Here's a table for all the mRNA's codes and the amino acids they code for. Each codon codes for a specific amino acid. Notice that some of the codons are special, like AUG. AUG is the start codon, which initiates translation by coding for methane. In this two-step process, DNA is transcribed into an mRNA, and the mRNA is translated into a protein.